So in today's class, essentially we will be talking about subcellular fraction using differential centrifugation. This is in your syllabus. So it is very important for a biochemist or a cell biologist to separate different cell organelles. So you have one cell, the cell has got organelles like nuclei, ribosomes, mitochondria, and different cellular organelles there. And you want to separate them, you want to study them separately. And for that you need to isolate mitochondria, you need to isolate ribosomes, you need to isolate the nuclei, you need to isolate individual organelles, and then you can do a study, whatever you want. You have to enrich them. So you need to break open the cell and separate them, but since they are so small, breaking them open, you can make everything into a paste, or you can basically sort of destroy everything, or it could very well happen that all the DNA inside your nucleus, it comes out and it sticks everything together, so you cannot separate organelles as such. So this problem exists. তোমরা শুনতে পাচ্ছ তো?
Yes, sir.
Yes, sir.
কেন আমার লোড শেডিং হয়েছে এখানে?
হ্যাঁ স্যার,
Okay.
আমার
হ্যাঁ, so I'll share my screen. এসছে, এটা দেখতে পাচ্ছ সাবসেলুলার ফ্র্যাকশনেশন?
Yes, sir.
আমি
Yes,
আজ
sir.
আমার তো পুরো লাইনাক্স সিস্টেম, আমার ডেস্কটপটা লাইনাক্স সিস্টেম হয়ে গেছে, উইন্ডোজটা করাপ্ট হয়ে গেছে, ওইটাতেই করতে হবে, কিছু করার নেই। আচ্ছা, so the topic today is subcellular fractionation. So like I told you, what is the importance of separating subcellular fractionation? So what is why do you want to break it up? The first thing is that if you want to study a particular cellular component, we need the sample in large amount. So you want to study mitochondria, you need lots of mitochondria, and that the mitochondria has to be intact. So ফেটে যাওয়া mitochondria চলবে না। And if for example, if your chromatin comes out from the nucleus, it's going to stick all the organelles with it, so it is basically you destroy the cell as totally, you will not get the organelles also. So this technique enables us to separate pure samples of subcellular components in reasonably active states. You want the mitochondria to be in a working condition, not that you have isolated it and then this particular mitochondria is damaged, you have holes in the membranes, and therefore you can't study the electron transport chain. So you need to isolate these mitochondria in a working condition. And it is a successful approach for a large number of proteomic studies. So you want to study about mitochondrial proteins. So how do you separate? So you first need to have the mitochondria pure solution of mitochondria with you, and that pure batch of mitochondria, and there you can break and you can isolate the proteins, and then you can say that these are the mitochondrial proteins. You can study them. Then you want to study metabolic activities, say for example studying the electron transport chain on mitochondria, so you need to isolate the mitochondria as whole. And so there is basically two types of techniques for separation of organelles. And first is differential velocity, this differential centrifugation, and the other is density gradient centrifugation. In your syllabus you don't have density gradient centrifugation, but you have differential velocity centrifugation. So what is basically, just a brief introduction, what is density gradient centrifugation? What it does, it separates subcellular components based on their density. So mitochondria has got a different density. You take 1 gram of cells, say liver cells. Your the mitochondria of all the liver cells, they have a density. The nucleus has got a different density. Ribosomes have got a different density. So everybody has got a different density. So in a tube, if you make a non-ionic molecule like sucrose, glycerol, gradient, that is, gradient means you have, say, for example, the highest concentration, 10% sucrose below. On top of 10%, you have 9%, then 8%, 7%. You have a continuous gradient. You make a continuous gradient. That is the 7, 8, 9, 10. These are discontinuous. It's like 9 এর থেকে একটু কম, then তার থেকে একটু কম. So gradually, as you go up, the density decreases till you have a 2% to 10%. You can make a gradient. You can also have a step gradient. That is, first you pass them through a high density, the centrifugation. Then you pass them through a medium density, then you pass them through a low density. So you can do this also step by step. You can do this. Okay, শুনতে পাচ্ছ তো?
Yes, sir.
Okay.
Yes, sir.
So centrifugation means you rotate them at high speed. If you rotate them at high speed, you will see. So this is the sample, your cell extract. This is a sucrose gradient. And below you have the highest gradient, and gradually it becomes low till the high. The blue color becomes less gradually. And then you centrifuge, you rotate them at high speed. There are specific speeds, 5 to 20% sucrose. And then you will have or at according to their density, the cellular fractions will separate. You can have a fast sedimenting component which will go to the higher density, a slow sedimenting component which will come float at a lower density. For example, lighter components, they will not be able to penetrate through high gradient, and heavier components, they can penetrate. They will not float on lighter things. ভারী জিনিস ডুবে যাবে, হালকা জিনিস ডুববে না, something like this. So you can have this. You can separate components like this, and then you can pierce the tube and you can take out all the components from the different fractions, different layers. You can take the respective components and you can see which layer has got which component. Perhaps this has got the mitochondria, this has got the nucleus. This is how you separate on the basis of density. The nucleus will sediment at a density same as that of for nucleus at this point, and maybe mitochondria will be at this point. This is how the separations take place. Now you have differential velocity centrifugation. So this separates cellular components based on size and shape and the density. Larger, more dense components will pack at the bottom of the centrifuge tube faster and at lower speeds than smaller, less dense ones. So at lower speeds, more dense components will go below faster. The smaller ones will go at a lower speed, they will go slower. ওরা ঢুকতে পারছে না because of the density. So initially the total cell extract is centrifuged at a low speed for a short time to remove unbroken cells. Later, the speed and time of centrifugation মানে ঘোরানো are progressively increased. So first slow speed একটা ভাগ করে দিলে, তারপরে একটু আস্তে আস্তে স্পিডটা বাড়ালে গিয়ে এক একটা করে ফ্র্যাকশন at each step you are separating each fraction at each step. So the final supernatant after stepwise removal of nuclei, mitochondria, vacuole, ribosomes is called the soluble fraction. It contains soluble proteins and other small components of the cytosol such as TRN. Let's see how they do it. So এটা দেখি একটু বড় যদি করতে পারি. Are you able to see this? Cut leaf tissues, liver, heart.
Yes, sir.
দেখতে পাচ্ছ
Yes,
তো?
sir. Yes, sir. Yes, sir.
Okay, okay. So what you do is you first step is you cut the leaf tissue, heart, liver in ice cold buffer. So chota chota kuchi 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 kuchi, you just chop it in a buffer. Cold to you use cold, so other the protease which will destroy things, it is stopped. You can even add some protease inhibitor, or you can grind the tissue in a mixer, Waring blender. In a mixer you can chop the cells. After chopping, you do it like this, then you filter it in a muslin cloth, and you have the cell extract in a tube. The first at a low speed, 1000 into G. This is a speed for 10 minutes. If you circulate it, the pellet, this pellet is there. This pellet is taken. This supernatant is the top liquid part, and the pellet is formed at bottom. This pellet will have the nuclei. So you have a nuclei enriched fraction. Now you take the supernatant, and again you do it at an increased speed, 3000 G for 30 minutes. What happens? একটু স্পিড বাড়িয়ে দিলে, this is at 10,000 G. 10,000 G, যেটা 1000 G ছিল, 10 গুণ স্পিড বাড়িয়ে দিলে, 10 folds. And now you have in the pellet mitochondria and chloroplast, they come over here. So this fraction is taken away, and the supernatant is again taken, and the supernatant at 100,000 G dilo. So the pellet, in the pellet you'll have endoplasmic reticulum, Golgi, and other membrane fragments. You will get them. So in the first fragment, you will pellet, you will have nuclei. Then you have separated mitochondria, chloroplast. Then you have separated Golgi, and other membranes. And then at 300,000 G for 3 hours, you will pellet the ribosomes. ঠিক আছে? And then the supernatant is now organelle free cytoplasm. You'll have some tRNA, some soluble cells, super cytosolic components. So you have the pellet one, low speed centrifugation, whole cell nuclei and cytoskeleton will fall. Then again medium speed you do it, mitochondria, lysosomes, peroxisomes will form. Then high speed centrifugation, you have microsomes and other small vesicles, Golgi body and all, they will come out. And very high speed ultracentrifugation, you have ribosomes and very large molecules which will come out. Clear?
Yes, sir.
তুমি এটাকে ঘোরাচ্ছ, you are rotating them at very high speeds. We will I will one day I will take you what is G speed, gravitation into gravitation speed, sweat work unit. You will understand this, but this is how you separate them. So basically the procedure is simple. Homogenizing buffer has got sucrose. You can use other components also over here. EDTA will stop the proteases, and you use a buffer. Tris HCl is a buffer. It will change the pH. It will resist changes in pH. So you homogenize mortar pestle, homogenize korle, centrifuge korle, you pellet the nuclei, the supernatant 15,000 G for 15 minutes, then the 1,000 G for 10 minutes. The same thing, and then you supernet the cytosol. This is how you actually do the differential centrifugation. So it is clear today's class এগুলো clear হয়েছে তো?
Yes, sir.
How you separate them, different fractions. এইটা একটা আসবে, I think you can expect a 5 marks question. এই ছবিটা দিয়ে একটু লিখতে হবে।
Okay, sir.
I think this is a good way to you can expect some questions. ঠিক আছে, আজকে ক্লাস এখানে শেষ করছি। আবার আমরা দেখা করব ফ্রাইডে ইভিনিং।
Okay, sir.
Okay, sir.
ফ্রাইডে ইভিনিং বা স্যাটারডে ইভিনিং we will see.
Okay, sir.
Okay.
স্যার একটু
Okay, sir.
টপিকস গুলো বলবেন না, ইম্পর্টেন্ট যেগুলো আসতে পারে এক্সামসে?
আগে তো হোক, তারপরে বলবো
হ্যাঁ
ইম্পর্টেন্ট
স্যার,
টপিক।
একদম একদম স্যার, ঠিক আছে।
এবারে শেষ করার চেষ্টা করছি পুরোটা, কারণ
Okay,
এবারে
sir.
পরীক্ষা তো পুরো ফুল ফ্লেজেড ভাবেই হবে। হ্যাঁ, এবারে আগের বারের মতন টুকে বের করতে পারবে না, আর এবারে আমরাও ইয়ে দেখবো না খাতা। হ্যাঁ, তো এবারে বাইরের টিচার দেখবে, সুতরাং এবারে একটু ভালো করেই করতে হবে। এই যে সব পাঠাচ্ছি এগুলো লিখে ফেলো ফাটাফাট।
ওকে স্যার।
হ্যাঁ, নয়তো ঝাড় খেয়ে যাবে। ঠিক আছে, টাটা।
বাই স্যার।
স্যার।
ওকে।